Okay, so I have drawn out for you the overview of cellular respiration, glycolysis, which is the splitting of glucose into pyruvate. If there's no oxygen, that pyruvate will go over into fermentation, which also takes place in the cytoplasm without oxygen. And it will depend on whether you're animal cells and you go through alcoholic fermentation. If you're an animal cell, you'll go through lactic acid fermentation to make lactic acid. If you are a yeast cell, you'll go through alcoholic fermentation to make ethanol. And the whole purpose of fermentation is to make NAD plus, to regenerate your NAD plus so that you can continue to do glycolysis, which will give you a net output of 2 ATP, right? Now we're going to talk about what happens when pyruvate will enter the mitochondria if there's oxygen present, and we're going to draw that out. So I'll share my screen. Oh, no. I accidentally just tried to share my phone, which would be weird. All right, so let's try this. Screen share. Oh, interesting. Plug-in required. Hold on. Hold, please. Okay. Okay, there we go. Sorry. Okay, so we've done glycolysis and fermentation. And now what we're going to be drawing out is, oops, I'm going to make it a little bigger there. We're going to be drawing out pyruvate oxidation, right? We know that oxidation, so oil rig, so oxidation is loss of electrons or loss of a hydrogen. So pyruvate oxidation and the Krebs cycle, right? Sometimes called the citric acid cycle. And I'll try to be clear on when I use those. Let's talk again about the overview, when you want to do this. So first thing, this occurs only in aerobic respiration, which again means when there's oxygen present, right? When there's oxygen present, what will happen is pyruvate enters mitochondrial matrix, so the most inside part of the mitochondria, right? And those are the two main things that you need to know. Let's talk quickly about the ins and the outs, right? So the ins for pyruvate oxidation and the Krebs cycle are we going to put 2 pyruvate, right? And remember, pyruvate is a 3-carbon molecule. We're going to say we're going to have 4 NAD plus. But remember, since we have 2 pyruvate, this is going to be times 2. 1 FAD, and that's going to be times 2. The outs are going to be 1 ATP, but we're going to do that twice because we have 2 pyruvates. We're going to get 3 carbon dioxide times 2. We're going to get 4 NADH times 2, and we're going to get 1 FADH2 times 2. All right, so those are kind of the ins and the outs, and we're not really going to talk about a net for pyruvate oxidation and Krebs cycle, but what's really important are this is going to go to waste, right? These 2 are going to be sent over to the electron transport chain, right? Those are the main generators of those. But if you'll notice from before, glycolysis also gave us 2 NADH, which can also be sent to the electron transport chain. All right, so let's start drawing this out. So pyruvate came in, and I'm going to draw a little mitochondria here. So here we have the outer mitochondrial membrane, and we're going to have the inner mitochondrial membrane, right? All of this is going to go into the matrix. So this pyruvate, which is again our 3-carbon sugar, that's going to be our pyruvate that came in from glycolysis, and that's only when we have oxygen present is it going to be doing that, right? And there are transport molecules that are required for getting pyruvate across those two membranes, right? And we talked a lot about types of membrane transport proteins, right? So this pyruvate is going to be first converted. So it's going to be converted to a molecule that has 2 carbons plus a CoA, all right? And that is called, this enzyme, I'm gonna modify this just a little bit so I have a little more room. This is called acetyl-CoA, right? So, whoops. So we went from pyruvate, which is a 3-carbon sugar, to acetyl-CoA, which is a 2-carbon sugar. So what happened to that extra carbon, all right? So what we're gonna see is that we put in, so the thing that happened to that carbon is we're gonna get carbon dioxide out. So one of those carbons was converted or removed into a carbon dioxide, right? We also will see, I'm gonna drop this way, is that we're gonna put NAD plus in, and we're gonna get NADH out, right? Just to keep in mind that every time we're gonna do this, for one glycolysis, we get 2 pyruvate. So this is gonna happen 2 times, basically, right? The other odd thing that we have to put in to this reaction is called coenzyme A, right? And that's where we get this CoA being added to the acetyl-CoA, all right? So that process there that I just drew out is called pyruvate oxidation, right? So this process here is called pyruvate oxidation. And you should know that it is being oxidized, and at the same time, NAD plus is being reduced, right? So that's a redox reaction. So that's pyruvate oxidation, and that happens in the matrix. Now, we're gonna take this acetyl-CoA and we're going to bring it into the Krebs cycle, right? So acetyl-CoA is going to come in, and in the Krebs cycle, and I'm actually gonna, again, I'm gonna make this a little bit bigger. So I apologize if this is causing you guys trouble on your end. So we're gonna make those cristae of the mitochondria real skinny over here so that we can show you the Krebs cycle. All right, so when acetyl-CoA comes into the citric acid cycle, coenzyme A actually gets kind of kicked off. And what we're gonna see is that when it comes in, it is gonna be added to a 4-carbon molecule, right? So if we have 4 carbons and we're adding 2 from acetyl-CoA, how many is that gonna give us? That's gonna give us a 6-carbon molecule, right? And what we're gonna see in this whole Krebs cycle is that we're going to see that we're gonna lose carbon dioxides. So in this first step, we're gonna have 1, 2, 3, 4, 5, right? So what happened there? You will see that we, whoops, where did my little pen go? Sorry. There we go. What you will see down here, is that as we are going to lose a carbon, we are going to get carbon dioxide coming off, right? Oops, sorry, I'm looking at my face. Hopefully you can see that. We're going to lose a carbon. Here, we added 2 carbons from acetyl-CoA. Right? So here we got rid of a carbon, and now we're down to 5. At that same time, as we oxidize that molecule, what we're going to see is that we're going to, again, reduce NAD plus, right? Then, as we go on here, we're going to go from 5-carbon molecule, and we are going to go down to 1, 2, 3, 4 carbons. So what did we do here again? We're going to see that one of those carbons went away, and we're going to get carbon dioxide off as waste. And at the same time, we are going to reduce NAD plus in order to give you NADH, right? So we've already gotten 2 carbon dioxides released, and then there's going to be multiple enzymes here. And we're going to have 1, 2, 3, 4. We're going to get a bunch of enzymes, and we're not going to talk about all of them, but that 4 carbon molecule is going to be converted to a different 4 carbon molecule. And in the process, we're going to see several things happen, right? We're going to see that, and you don't have to know the steps here, but we're going to see, oops, skinny pen. We're going to see again, and NAD plus be reduced to NADH. We are going to see an FAD be reduced to FADH2, right? And we are going to see that ADP is going to turn into ATP, right? So during the Krebs cycle, so this is the Krebs cycle, what we see happen in the Krebs cycle is that we are going to input acetyl-CoA. It is going to join with a 4 carbon molecule to make a 6 carbon. We're going to lose carbon dioxide. We're going to lose another carbon dioxide. And in this whole process, what you're going to see is that you're going to generate. So in the Krebs, you're going to generate NADH and FADH2, which are going to go to the ETC, and you get a little ATP out, right, as well. And you're going to get CO2 out, right? All right, so that is pyruvate oxidation and the Krebs cycle. So pyruvate oxidation, where we're oxidizing pyruvate to give you this acetyl-CoA. Krebs cycle, acetyl-A comes in, and you are going through this cycle of kind of oxidizing that 6 carbon molecule, and you're going to get carbon dioxide out and NADH and FADH2 out and a little ATP, right? All right, so now we are going to go to the next and final part of cellular respiration, which is going to be the electron transport chain, which is called the ETC, and chemiosmosis, right? And that's probably the hardest conceptual thing that we're going to be talking about in cellular respiration. All right, so again, let's just talk about some of the key points here. So first off, this is going to be in the mitochondria, and in particular on the cristae, or those membrane folds. So it's going to be on the inner mitochondrial membrane, right? We are going to see that we need oxygen for this to happen. So this is going to be aerobic respiration. Sorry if I spelled that wrong. That's really painful for me. This is going to be, and what we're going to see about this oxygen that's really important is that it is the final electron acceptor, right, in the ETC. That's why we need oxygen for this process. It's the final electron acceptor. And it's going to accept electrons and create water, which is what we're going to see. All right, final electron acceptor to make water, to make H2O. And the last thing is that we're going to see a proton gradient build up in the intermembrane space, right? So that's why understanding your structure of the mitochondria is so important, because you have to understand where the matrix is and where the intermembrane space is and where the cristae, so you can kind of put all of these together in your mind, all right? Okay, so what we are going to see is that we are going to have the input. Well, let's do the inputs and outputs again. All right, so in and out. So the inputs are we're going to have NADH, right? And we are going to have, here, I'm going to scoot that over a little bit, actually. Hold on. Okay, so the inputs are going to be NADH, all right? And we are going to also have an input of FADH2, right? So for the NADH, we're going to have 2 from glycolysis, right? We're going to have 2 from pyruvate oxidation, and we're going to get 6 from the Krebs cycle. So we have a bunch coming in, right? For FADH2, we're going to get 2 from Krebs, 2 from the Krebs cycle, and that's it, right? And those are all going to come from, okay, never mind. And then the outs. So what we're going to see coming out of the ETC and the electron transport chain is that we are going to get about, and this is completely an estimate, about 34 ATPs, and we're going to get water out, right? And in the process, we're going to see some things get oxidized. So this whole process here, the ETC and the electron transport chain, is called oxidative phosphorylation. Oxidative phosphorylation. Okay, so I'm going to try to figure out how I want to draw this out. All right, so what we're going to see is that I'm going to just draw a kind of plasma membrane here, right? And I just want you to remember that when I draw a plasma membrane, I'm really drawing, it's really a lipid bilayer, right? So we're going to have a membrane there. And this is going to be the kind of the cristae folds, right? And I'm going to have the matrix up here, the matrix there, and I'm going to have the intermembrane space here. Intermembrane space. And then I want you to remember that there would also be another membrane here, right? That would be kind of the, this way. That would be the membrane, the outer membrane of the mitochondria. Outer membrane of the mitochondria, right? So out here would be the cytosol, for example, right? And inside would be the matrix. Okay, so in the electron transport chain, we're gonna see 1, 2, 3, 4, 5. We're gonna see 6 different proteins. And I'm gonna just draw those here in purple. So we have, and you don't have to know the details of all of these, but we're gonna see protein there. We're gonna see 2 proteins here. See another protein here. 1, 2, 3, 4, and then we're gonna see 2 more proteins. All right, so these are a lot of membranes that are on the cristae of the mitochondria. And then we're going to see a special one here. That looks kind of like a little lollipop or a mushroom or something like that. All right, and that's going to be the real special one called ATP synthase. Okay, so as these NADH, so we have NADH and FADH2 that are going to come in. FADH2 that are going to come into this system, right? So NADH is going to come in and become oxidized, right? When it becomes oxidized, it is going to donate electrons into the electron transport chain, right? Those electrons are going to be moved along the pathway. So they're going to move along, they're going to go here, they're going to jump here, they're going to go here, and they're going to go here, right? So they're going to move along that pathway, which is called the electron transport chain, because those electrons donated from NADH are going to move along this chain, right? Similarly, we're going to see something very similar where FADH2 is also going to donate electrons to the electron transport chain, and in the process, it's going to be oxidized to FAD. And those then are also going to move along the electron transport chain, right? You may ask, what is happening to these electrons, right? So it turns out that these electrons are going to then come out of the electron transport chain, right? And we are going to see that these two electrons, right, 2 electrons at a time, are going to combine with half in oxygen. I'm gonna actually draw this a little bit more clear so you can see this. So the two electrons are going to combine with half in oxygen, and that is going to come together to create water, right? So that means that oxygen is the final electron acceptor, and that's going to create water at the end of the ETC. That's why you have to have oxygen. So if, for example, you blocked this pathway somehow and didn't have an oxygen, these electrons would build up. You couldn't then have more NADH and FADH2 coming in, and the whole system would fail to create that gradient that we're going to talk about now. All right. So those are the flow of electrons. So we know electrons are full of energy, right? So something really cool happens in this electron transport chain, right? So what we're going to see is that as these electrons get moved from one protein in the electron transport chain to another, we're going to see that H's get pumped in to this to this intermembrane space, right? You don't have to know exactly what molecules it happens on, but what you do have to know is that as electrons are moved, H's get pumped into this intermembrane space, right? Intermembrane space of the mitochondria. And these H's will build up an electrochemical gradient, which we've already talked about, and we know the energy can be stored in electrochemical gradient, right? So what's really cool is that now these H's that have this electrochemical gradient can flow down their gradient through this amazing molecule called ATP synthase, right? And it is this amazing rotor machine that uses the power of those H's flowing down to, so these H's flow down their system, and at the same time what's happening is that they take ADP and inorganic phosphate to create ATP, right? And that's how we're going to generate our ATP, right? And what we'll do here, so again, NADH and FADH2 are becoming oxidized, hence the oxidative part. They're going to feed electrons into the electron transport chain. At the same time, you're going to pump hydrogens into the intermembrane space. Those hydrogens are going to flow down their concentration gradient through ATP synthase, which is going to generate ATP. It's really important that you know that we have oxygen as its final electron acceptor through that ETC to create water. All right, so couple things I want to mention here. This is all about kind of the theoretical yield, and I would not test you on this, but I think it's so cool. So theoretical yield. All right, so this is kind of the theoretical yield. So if you have one NADH through the electron transport chain, you're supposed to make about 3 ATP. For one FADH2, you're supposed to make about 2 ATP. And that makes sense because the FADH starts later in the pathway, right? So there's less hydrogen build up, meaning less ATP. All right, So in this process, how many NADHs did we, whoa, sorry, my iPad just stopped working. Okay, so let's go back and look at how many NADHs we had total. So we had 6 from Krebs, 2 from pyruvate oxidation, and 2 from glycolysis, right? So that's 10, right? So if we had, for example, 10 NADH, so we starting with, if we have 10 NADH, that's in theory about 30 ATP, right? We have 2 FADH2, which would give you about 4 ATP. And this is why people always say that oxidative phosphorylation gives you about 34 ATP, right? And then we also have 2 ATP from glycolysis, and we have 2 ATP from Krebs. So that's how you get your total of 38 ATP. So just so you get a sense of where that number is coming from. All right, so that is the drawing of both of pyruvate oxidation, Krebs cycle, ETC, and chemiosmosis. I guess I didn't mention exactly what chemiosmosis is. And chemiosmosis, and I don't love the name, but chemiosmosis is the process of H's flowing down their concentration gradients to make ATP, right? And we know that happens through this cool enzyme ATP synthase. All right. Okay, I will stop there.